Hey everyone, welcome to This Explainer. Today, we're getting right inside the examiner's head. We're going to decode exactly how to bump that B up to an A star for Cambridge Biology topic 4.2, which is, of course, simple and facilitated diffusion. You know the difference between those two grades? It literally just comes down to how well you understand the specific demands of this one topic. So I'm going to show you exactly what they're looking for and how to grab every single mark on the table. Okay, let's jump right in by exposing one really tiny mistake. This thing costs students crucial grade boundaries before they even realize what's happening. You probably think you know exactly how diffusion works, right? But Cambridge examiners, they have a highly specific set of criteria. They want to see connected, conceptual understanding that you can apply to totally weird, unfamiliar contexts. And honestly, there is a massive trap just waiting for you right at the start of your paper. Listen to this direct alert straight from the examiners themselves. It says, students lose marks by not showing working. Even if your final answer is wrong, method marks can save you. So your standard revision totally fails if you aren't securing these process-driven method marks. Think about it. Even if your final calculation goes completely sideways, writing down your method is your ultimate safety net. It doesn't matter how well you memorize the textbook, you have to literally prove your analytical depth by putting your whole process right there on the page. Section 1, Core Diffusion Principles. Let's lock this down. So, securing your baseline marks, that requires ultra-precise biological definitions, not just some superficial summary. You literally can't just write things move. No way. You need to explain that simple diffusion happens directly through the phospholipid bilayer, whereas facilitated diffusion, that requires channel or carrier proteins. Academic benchmarks demand logical reasoning here. You've got to explain exactly how and why molecules cross that membrane to get full credit. Now, principal examiner reports consistently highlight a massive trend among top candidates. They make cross-topic connections. To hit that A-star tier, you just can't view diffusion in a vacuum. It's like a giant puzzle. You need to link it directly to cell membrane structure, connect it over to water potential, and relate it to active transport mechanisms. That holistic, big-picture understanding, that is exactly what gets you top marks. Moving right along to Section 2, the procedural framework. Okay, in just a second, I'm going to reveal an examiner clue that completely changes how you answer. But actually, hold on. First, you need the framework that makes that clue super obvious. Because without a systematic approach, interpreting complex data and graphs under massive time pressure, it is incredibly difficult. You absolutely need a system. So here is the exact step-by-step sequence to guarantee you process stimulus materials systematically without missing a single beat. Step 1, deconstruct the question. Step 2, extract the relevant data. Step 3, select your analytical framework. Then step 4, and guys, this is absolutely crucial, execute with clear notation, because this is exactly where those method marks live. And finally, step 5, present your answer with precise biological terminology. Now when you extract your data, you have to know exactly what to do with it. You need to instantly pair the correct statistical analysis with your biological data types. So you've got your t-test for comparing means, chi-squared for categorical data, and correlation for variable relationships. Honestly, matching the right test to the right data is a completely non-negotiable skill for topic 4.2. You've just got to know it. All right, let's hit section 3, spotting the errors. Let's break down a quick comparison. A seriously flawed answer lacks context, has 0 units, and the reasoning just totally falls apart. But a top band answer, that elevates from a B to an A star because it uses strict, correct terminology, shows incredibly clear step-by-step working, and deeply evaluates the limitations of the evidence. Basically, the flawed approach is a wild guess, while the top band approach is a fully justified scientific recommendation. Here's a really brilliant, quick memory hack straight from subject specialists. Build your own mnemonic using the first letters of those key terms. You know, when you're sitting in the exam hall and that timer is ticking down, the pressure can literally make your mind go blank. Having a customized mnemonic ensures you never drop those essential, mark-yielding terms when you're trying to explain the mechanics of simple and facilitated diffusion. Okay, almost there. Section 4, Top Examiner Warnings. So there are exactly 3 major mistakes that examiners see year after year on topic 4.2. These are the specific errors that are unnecessarily capping your grades, and we are going to fix them right now so you don't fall into the exact same traps. Let's dive into this checklist. Number 1, forgetting to include units in calculations. That is literally throwing away easy marks. Number 2, failing to double-check those units and terminology before you turn the page. And number 3, writing generic textbook definitions instead of perfectly aligning your answer to the highly specific biological context given in the prompt.
Context is everything.
But wait, what if you know the biology perfectly? You show all your working, you include your units, but a single command word completely caps your mark band. Yeah, mastering diffusion just isn't enough if you don't know exactly what words like explain, describe, or evaluate are secretly asking you to do. Misinterpreting those command words is the hidden cause of massive mark loss. To decode those exact command words and secure your top mark band, definitely subscribe to our YouTube channel Ace the Exams at acetheexams_IGCS and go practice those past papers. But before you go, I want to leave you with this one thought. Are you studying just to blindly memorize a textbook, or are you studying to actually master the examiner's mindset? Implement this framework and I'll see you in the next explainer.